Okay, welcome everybody to the College of Optical Sciences Colloquium. It's a great pleasure for me to introduce today's colloquium speaker, Professor Raymond Kostuk. Ray is a professor here at the University of Arizona, as most of you know, and he has a joint appointment in ECE, I think that's his home department, and the College of Optical Sciences. Ray received his BS in Physics and Math from the U.S. Coast Guard Academy in 1972 and served for 10 years as an officer in the U.S. Coast Guard. I didn't know that. Then he studied at the University of Rochester, where he got his MS in optical engineering in 1977. He then went on to Stanford, where he got his PhD in electrical engineering. And after his PhD, he went to IBM, working on optical storage problems. And after that, he became a professor here at the University of Arizona in 1987. His primary area of expertise is holographic concepts, materials, and applications. And yes, he is a fellow of the Optical Society of America and SPIE. And I believe today's talk will be titled Adventures of a U.S. Coast Guard Officer.
Yeah.
Or maybe not.
Okay.
Or holographic applications in medical imaging and photovoltaic systems. Thank you very much.
Thank you, Rolf. Can you hear me okay? So one thing Rolf didn't mention is when I was in the Coast Guard, my sea duty was on an icebreaker. So here I am in the desert, and I spent the last 30 years here. So anyway, I have great opportunity. I'm actually on sabbatical this semester, but I'm more than happy to come and talk about our work. I really enjoy working with lots of good people. As Colton mentioned, I need to add a few more names. I probably do. But these people here have helped me specifically in the two areas I want to talk about. These are very diverse areas, but they've kind of occupied my main interest and work for about the last 10 years. And so we're going to talk about medical imaging and solar energy. So, trying to save the world in many ways, in at least two ways. But I have a great interest in holography. It's been a kind of a passion of mine. I think I made my first holograms in 1974, so going back about 40 years. When I was in the Coast Guard, I wanted to do something. I was stationed in Seattle. I found a group at University of Washington that was doing biomedical research, and they were actually making holograms of cow's eyes to kind of map the vitreous humor and the retina and so forth. So we would go down to the stockyards and collect cow's eyes and make holograms of them. Anyway, let me move on because I'm sure I'll run out of time. I have some great sponsors as well, NIH, NSF, and Department of Energy. So what I'll do today, give a brief overview of holography. Some of you may or may not. I invited some undergrad students here, so they may not have a great background in that, so I'll give a little bit of an overview. And then I'll talk about these two projects. One is a volume holographic imaging for the detection of ovarian cancer in particular, and also the applications in photovoltaic energy systems. So next fall, I'll resume teaching my holography class, 527. Please, if you're interested in this area, you're welcome to come and see it and come and take it. So this is a picture of one of my favorite holograms. You're welcome to come and see it afterwards. I brought it here. I would, if you have a chance, have a look at it. I guess I could pass it around. You might need a cell phone light or something, but it's really striking, I think, and it shows the kind of the unique things about holography and some of the captivating nature of holography, this three-dimensional aspect to it. It's much different from a normal photograph, of course, and I think it's even different than computer graphic rendering in a sense, because here you have stored this information in one snapshot of a phase information about an optical field. So this is something that's unique and very useful and interesting about holography. So let's first do a little lesson here. So to make a hologram of this type, what you first do is you need to store the phase. So to do that, you have to do interferometry or some type of interference effect. And so here we have a reference beam, an object beam forming our fringes. We then insert some type of recording material in the fringes. Now, the recording material could be a film. It could also be a digital camera. There's a big area of digital holography, which is for microscopy and so forth. There's a huge area. Actually, Joe Goodman made the first ones back in 1967, 50 years ago. So 256 pixels on the camera. So at any rate, so you put a recording material there, and then you can reconstruct the object beam either with the original reference beam, or you can use a conjugate wave to reconstruct the conjugate of the object wave. Both of these are useful. I actually mostly use this one for the reconstruction process because I mainly do holographic optical elements. So the hologram is a grating. That grating is a set of scattering, periodically spaced scattering sites, which could be a modulation in either the absorption or conductivity or the phase through a path length or refractive index and depth modulation or change. Primarily, or I should say that phase gratings, though, are much more desirable for the things that I do in holographic optical elements because you can get very high diffraction efficiency for these elements. Now, one way to get this high efficiency is to make what's called a volume hologram or a volume grating. And so this has a set of conditions on the physical parameters. For instance, the period is very close to the wavelength. The period is also much less than the thickness of the grating. So this creates basically channels here so that only the diffracted order can make it through. One diffracted order at that, not multiple diffractive orders. The other ones get dephased in passing through this, and so you can get all of the light diffracted into pretty much a single diffraction order. And this property is called Bragg selectivity, where you're, again, producing this single beam with high efficiency. And it's actually quite similar to a one-dimensional photonic bandgap element. All right. So some properties that we'll use of the hologram are the selectivity. And so here, this is the angle and diffraction efficiency, which is diffracted light over incident. I just threw in some numbers in one of my programs here to show what you can get. And so here we have a fairly broad angular field of view for the hologram. And we get that by decreasing the thickness of the grating. Or we can make it much thicker, get it much more selective, so there's a very small channel, again, that you can only pass through, and you can narrow that up. So in these two applications, we'll use this property, and diversely. Okay, so one application uses one, and the other application uses the other. Another feature of holography is that you can multiplex or store many gratings in the same space. All right, so you can record these either simultaneously, where you send several object beams in with a single reference beam, and then during reconstruction you generate all of these object beams, or you can do it sequentially, where you code a different reference beam angle with a specific object beam. So this type of multiplexing is quite useful for broadcasting information. When I started my holography with Joe Goodman, we were looking at optical interconnects, so we were looking at broadcasting information, clock signals, and so forth, and for chips. And here, this is perhaps more useful for things like data storage, and it's also the process that we'll use in the medical imaging application. Another feature of holography is dispersion. So this is the grating equation here. And so you can see, if we have a fixed period, that the red light's going to be diffracted more with a greater angle here than the blue light. And here's a nice hologram that one of my former students, Shelby Vordran, had made and is holding here. And you can see a nice dispersive diffraction from the sunlight that's illuminating it. Okay, and So finally here, you can make a hologram again with simply with an interferometer, and so you can split your beam, set it up with call one of these an object beam and the other a reference beam. If you have two plane waves, it really doesn't matter. But you can also put stuff in the object beam, like in this hologram here. We have the woman's face. We could also put some beam shaping optics in the reference beam to modify that and, say, change the reference beam angle and so forth. So the point of this is it could be made, they can be made fairly inexpensively. And actually, this is one reason I really like holography, is because it's a great educational tool. You can make holograms with relatively simple equipment in a lab. I brought stuff in the back of my car and drove off to other universities and set it up and had students make holograms for an afternoon. So it's a very nice mechanism for teaching optics, various types of optics. So it's very useful in that regard as well. More towards these applications, you can make low-cost holograms in very large volumes. And this was demonstrated by Prism Solar Technologies. It's a company here in town. And they actually filed a patent on making a roller manufacturing system where they can make kilometers of this stuff and run it 24/7 for solar photovoltaic systems. And that's what you need. You need something for a lot of applications. You have to think: is this scalable? Is this something that I can mass produce? Is it necessary to mass produce it? In the solar case, it is. In the medical imaging case, not so much. Although we still want to keep costs down in that area if we ever expect it to become a useful instrument. And we can make this on a variety of surfaces as well: glass, plastic, mylar, and so forth. Can all be coated with a material and processed and formed. Okay. All right. So let's start looking at some applications. So the first one is what we call volume holographic spectral spatial imaging. All right. And specifically, we're looking at ovarian cancer. Now, I borrowed this slide from Jennifer Barton, my colleague over at Bio 5. And so the thing about ovarian cancer, which is quite compelling, is that, again, it has a very high mortality rate. In fact, it's the highest mortality rate for gynecological cancers. And it spreads again, in 90% of diagnosed women. There's often no symptoms. There's very limited, if no, really, dominant screening tools that are available. And as you can see here, the 5-year survival rate for this cancer really decreases if it gets away from you. So, and the thing about it, again, it's asymptomatic, so you don't really know you have it. So why would you go get screened? There is a group of women who will get screened or tested. They are women who have a family history of ovarian cancer, so they're more apt to get screened. And if on the flip side of this, if a young woman gets screened, she would like to know, is this cancer developing or not? I'd like to have children. So, there's a lot of decisions that go into this, and so you need to have good tools for looking at it. Okay, so there is a real need for better detection techniques here. Okay, so again, you need to improve the detection of these hidden cancers, the occult cancers. We need instruments for guiding interoperative biopsy site selection, so you want to identify where the most suspicious areas are, and again, preserve fertility of premenopausal women if possible. Okay, so our effort in this started back around 2005, actually. George Barostatis and I were working on another project together, and Jennifer and I were also working on a parallel OCT system, which was coming to an end. So I was looking for something new that maybe we could apply holography to. And so George worked with Dave Brady and Dimitri Saltis and they wrote a really nice paper back in 1999, which is in IEEE Proceedings, on volume holographic imaging, multidimensional imaging that you can achieve with volume holography, and it kind of kicked off this area. So, I talked to George, and we started to develop this this program. So the whole feature here is that we're going to use this Bragg selectivity in this volume hologram in the imaging system. All right. Now, because the hologram, again, can diffract at specific angles, and it's also a dispersive element, we can get both spatial and spectral information from the sample. And so the way we're going to use it is we're going to actually select wavefronts that come from different depths within the tissue sample, and then use that multiplexing method to project these different wavefront information to different regions on a camera. So the idea of this is, keep in mind, is that the image quality of this system is not going to be better, say, than a confocal scanning system or an OCT system, which are other optical techniques, but it could potentially be a much cheaper way to go because we don't need to use scanning. So that's the thing to keep in mind. The image quality is not necessarily going to be better. It's comparable to a slit scan confocal system if you're aware of that system. But the whole purpose here is we want to try to develop a fairly low-cost system which can go out to a lot of clinics and give useful information. All right. Okay, so for the hologram, we need to have certain things. We need high Bragg selectivity to give us our depth selection, image contrast, and lateral resolution. We have this wavefront selectivity to do background rejection. We use the multiplexing to display multiple image depths. We use the dispersion to give us both the lateral field of view and for a form of fluorescence imaging in the system. And we can also incorporate into the hologram other filtering operations, such as phase contrast enhancement, all right, which was demonstrated by Yuan Lo, one of my former students. Okay, so we need this high selectivity. So we're going to need a almost like a crystal, all right? But the crystals are, like ferroelectric crystals are pretty expensive. So we've been looking at the last 15 years, actually. We've been working on holographic materials, one of which is this phenanthroquinone doped PMMA. So it's basically a doped plastic that we can form in a mold here. So our molding doesn't look actually too much different than this. We need to have a flexible system, a flexible mold, because the material, when it solidifies, all of this is solution. We pour it in here. It expands, all right, and if it was rigid, it would crack. So we put neoprene tubing here. We also add some mold release and some other things in it. And so what we combine is this, the monomer, AIBN, which is a thermal polymerizer, and phenanthroquinone, which is a photosensitizer. And so we basically bake this for 5 days at a 50 degrees temperature that's ramped up slowly to prevent bubbles and other issues with the formation. And then we cut this up, and we have our material. It's fairly thick, several millimeters thick. And this is going to give us the selectivity that we need. Here are some examples of the diffraction efficiency. And notice that the full width half maximum angular bandwidth is now hundredths of a degree, not angles, okay, not whole numbers or even tenths. We can also dope this. We've done some experiments doping it with nanoparticles to also enhance the diffusion process and the formation of the gratings, enhance the areas of high index, for instance. And so we can do actually nanoparticle manipulation in the polymer holographically with the exposure. So it's kind of an interesting area that we haven't really explored too much, but it's kind of a nice sideline. At any rate, we get this high selectivity, which we're going to need because we want to do this wavefront selection. So the basic operation of the system is hopefully diagrammed here. So here we have a a point source located, say, some distance away from the focus of this lens, it creates a slight wavefront curvature, and we record that with a planar reference beam at one angle here. All right? So now if we get light coming back from the tissue sample at that position, it's going to create that same wavefront. And now, because the grating vector has varied across the aperture of this hologram, it's going to diffract light and matches the specific wavefront. It's going to match light across the whole wavefront, and so we get a fairly strong diffraction of the light coming back from that location. If we instead come in here with a collimated beam, then that beam is only going to match near the center. So we will get a little bit of diffraction, but it's greatly diminished. All right? And so this is the wavefront selectivity. And these differences that we'll be looking at are on the order of tens of microns in the tissue sample. So that wavefront curvature is very small. Okay? And so that's why we need that high selectivity. So now we make a hologram, we multiplex it. Let's say we multiplex 2 gratings in there, and we illuminate the tissue sample with a laser. All right? If we do that, again, light comes back from all depths, but only these 2 depths at these locations where the holograms were recorded are diffracted with appreciable diffraction efficiency. And what we'll see on a camera, if we place back here, are 2 stripes corresponding to those 2 depths. Okay? So that's fine, but generally we like to see a little bit more field of view than just having a stripe. I mean, you could scan it with this with the laser, but we're trying to get away from scanning if we can. So if you take my class and you play around with these Bragg diagrams for a bit, you'll see, right, Colton? Yeah. You'll see here that if you have a grating vector, you can get high efficiency Bragg match. If you complete the vector diagram here, that means basically that you're going to get high diffraction efficiency. So you can see here that any of the propagation vectors along this bisector line here will satisfy that condition. All right? And so you can get a number of high efficiency diffractions occurring provided you change the angle. Okay? So this angle and wavelength duality is a very useful property. And in this case, what it allows you to do is, if you now spread the wavelengths of light laterally across that tissue sample, and now illuminate it with that kind of a structured illumination here, now what you'll get is a diffraction with a larger field of view, okay, from these 2 different depths. Okay, so this is a very preliminary system that we made years ago. So this shows, just to show how simple it is, it's basically, again, a hologram in a microscope. Here's the microscope objective. Here's the sample. There's a relay going to the hologram. In this case, we're just using side illumination. And then we have another relay and then the camera. All right? And so then again, if you multiplex these many holograms, you can get many different depths. So in this case, each separation here is about 75 microns, giving us a full scan or full depth range here of about 30 microns. This is illuminated with a 30 nanometer LED. And you can see here there is some resolution fall off. That's due primarily to the fact that we're recording these holograms at 514 nanometers and replaying them at 630. So there's a wavelength difference here. And again, if you take my class, you'll see that you get these chromatic aberrations that occur when you replay these without correcting for them. Okay? So, and the curvature that we're talking about here is about 300 micron in curvature. So it's pretty sensitive, but there is some ways around this, and we could still get useful information, and even with some of this image degradation that we're experiencing here. We can also get fluorescence imaging here. In this case, we illuminate with a 355 nanometer laser using an acridine orange dye. In this case, we only have 2 holograms here recorded, we get about 100 nanometers in spectral bandwidth. And you can see some of the features here. They change due to the different depths in that tissue sample. Okay, so our next step was to make a more professional version of this system that we can use actually in a ex-vivo study from our with our NIH program. And so we have a common source illumination here, or common path illumination, with Kohler illumination coming through. So here's where the sample would go here. The hologram, of course, is here. We have two light sources, so we can do both fluorescence and reflectance mode imaging in the system. We did a study with 26 patients. Ken Hatch, by the way, so Ken Hatch is the surgeon on our team that worked with us, and so we consulted with him a lot in the development of our instruments. And what he told us was that he only needed to see two depths. He wanted to see the surface, and he wanted to see several cell depths below the surface. So I did show you 5 holograms there that were recorded, but he said 2 is enough. So it makes life easier to multiplex 2 rather than 5 holograms. And so the images that you'll see are with 2 recorded holograms. So this is a kind of an example here showing the process. So here is the surface channel that we call a channel surface image, and here's the depth image. And these correspond to, again, these histological path or images here. These are actually samples that were cut out, prepared slides, and then they're stained. And this is the traditional way of doing it. It works great, but it takes a while to do. And so here we were just looking at about 50 microns below the surface. And you can see, again, changes in features, some of the features here. This was a fairly normal tissue sample here, in that it's fairly pebbled in structure, not a lot of coarse structure. This shows a more distinct variation. So here's the normal tissue, and here's the cancerous tissue. These are the histologically stained samples. And then this is the reflectance mode imagery. So you can see here this pebbled nature of the image for the normal tissue, and this more granular, irregular type shape for the cancerous tissue here. We also did, at the same time, so these are excised tissues which we put on our instrument. We also did the fluorescence imaging using just an applied membrane and nuclear staining dye. And so you can see additional edge features here with the fluorescence dye kind of added to the imaging process. So the next step in this was to actually see if we can make a handheld version of this that would be able to go into a surgical environment. All right. So several of my students, Isella Howlett and Michael Gordon and Wang Li Han, have been working on this this particular system. It's made out of off-the-shelf kind of components from Thorlabs. Here's the hologram. Here's a little camera that we can get from Thorlabs here. One of the things that we had, was most serious in the design was this probe. It's a grin rod probe, as shown here, and we were using common path illumination in that. So this probe goes into a plastic trocar, and so we had to go through sterilization process for this and clinical engineering certification. And so that was a real challenge. One of the biggest problems we had was the glue, the epoxy, the optical epoxies that we're using would kind of dissolve a bit in the process. So we had to do some more research, find better cements. There's an Epotech range of epoxies that work very well. We have that information if you're interested. It passed clinical certification, and Ken Hatch used it in 2 surgeries. The image quality is is okay. We still have a lot of issues with, some contrast issue here. Now, one thing that we recently, that Isella, she just finished her degree actually, and before I let her go, I made her publish this paper. But she came up with a nice idea of using, in this Grinrod system, there's axial chromatic aberration. And so rather than, again, having that as a detriment, we actually use that to an advantage. Because we now allow the axial chromatic aberration to bring light to focus at two different wavelengths. We sent two sources into this, a 660 nanometer and a 730 nanometer, 660 for the surface and 730 for depth, the longer wavelength, less scattering in the depth. And now the light comes back and is focused at the same point at both wavelengths. So the big advantage here is that we could use, instead of having a curved hologram, we can now use planar holograms. Planar holograms don't have this image degradation when you record at one wavelength and reconstruct at another. So it's a very nice idea that Isella developed and had for this design. Okay, so I've kind of been leading you astray here. I was talking about this wavefront selection that we have. However, in the system that we have used, we used an LED and we didn't really separate those wavelengths. So we're getting all wavelengths at all locations, which means that we have kind of a degenerate Bragg matching condition that occurs, which reduces the amount of background rejection that you get because you're not fully utilizing that wavefront selectivity of the hologram. Okay, so we looked at several ways of getting rid of that. You can use, of course, some image processing and so forth, and again, phase contrast, adding some phase contrast filtering, additional filtering to it. However, the real way to get rid of it is to use some type of structured illumination, or, God forbid, resort to a little bit of scanning. Okay, and so we're actually we looked at both of these. We used what we call confocal rainbow volume holographic imaging, which I'll explain in a minute. And Wang Li Han is finishing up some work using a swept source laser and a second grating in combination to do single axis scanning for this system. So for the confocal rainbow system, we basically bring the light in and use the hologram twice, one to disperse the light at the correct wavelengths, and then we have a perfectly matched coming back into the hologram to separate the two images. This actually works very well, although it's a real pain to align, unfortunately. And, well, another problem is when you go back and forth through that hologram, the quality of that hologram has got to be really good. Our quality is not that good. It's okay, but it's not good enough for that particular version. We're looking at some other variations of that as well. But you can see here the benefit of doing this is that now if you translate along the z-axis, you can see that there's a very sharp drop in the efficiency when you get away from the targeted peak efficiency value here. And what that translates into is background rejection and improvement in your contrast of the images. Okay. All right, so this is just a little summary here of looking at our system, this volume holographic imaging, with some other traditional methods, optical coherence tomography, confocal microscopy, and so forth. The main advantage, again, is, not that the image quality will be much better, or any better for that matter, but you could get it with no scanning, okay? And so that would provide a lower cost instrument, which with a less complex probe that you would need for the imaging process. Okay, so that's the first application. And now we're going to swing over to the other side and look at how we can apply this to solar energy conversion, all right? So we've actually looked at a lot of different applications within the photovoltaic conversion process. We've looked at, and even more than that. So certainly spectral filtering is something that we can do. We've developed a technique for ultra light trapping, which I'll explain. I started doing this in a collaboration with Prism Solar, Glenn Rosenberg, who actually took my class and in holography, and he went to work. He started a company years ago, TerraSun initially, and then that became Prism Solar. They started looking at these holographic planar concentrators. I'll explain that as well. So that got me started back in about 2000, about 10 years ago. Most of our work today has been focused on spectrum splitting, which, again, using these kind of nice properties of holograms to increase the efficiency of PV systems. We've applied it to microconcentrating arrays and also the photosynthesis active region, PAR, enhancement for filters for biofuel production. So we looked at, we had a San Diego power company came out, and we were looking at ways of enhancing biofuel production using holographic elements. Okay, so for this purpose, we need not real selective, but totally unselective, if possible. But anyway, a broad, broad angular selectivity and wavelength selectivity. Right? You want to capture a lot of the solar spectrum. The solar spectrum ranges. The useful range is from about 350 nanometers to 2 microns. So that's a huge range, okay, of wavelengths. So we need something that's very thin. And so we work on dichromated gelatin. We've been working on this for many years, probably 25 of my 30 years here. We've been working on dichromated gelatin. We have various processes for doing this. Our favorite right now is a simple mold process. We also have spin coating over in my lab and so forth for doing this. But this material is great, but it's a real pain to work with. You have to really control the environment and the parameters really well. And there's still a lot of mysteries in the process. But we can get pretty good results with it, and I'll explain some of these. So just for an example, we can get 2 to 3 micron layer thicknesses with delta N's of 0.1. Okay, so that's a huge index difference for a optical material, and this is something that we need. It's very similar to, like Angus MacLeod's thin film methods. There you have fairly high index modulations, high-low stacks of index-varied filters. We can do the same thing. So let me talk about some of the applications here. So we have, to start with, we have this holographic planar concentrator that I mentioned that kind of kicked me off with this with Glenn back in 2007. So the idea here, back then at least, silicon was very expensive. And so the idea of any concentrator is you want to replace expensive PV material with less expensive optical collection material that can channel that light down into that PV cell. So here the hologram collects light in these areas that are not covered by the PV cell and directs it over to the PV cell, either directly or through total internal reflection to the PV cell region. This element actually kind of looks like a little bit like a parabolic concentrator, compound parabolic concentrator, where you replace this parabola, this reflective parabola, with a hologram, and you can put it in a flat panel. PV is extremely cost sensitive. Anything that reduced, the big thing here is you want to reduce the dollar per watt or the dollar per kilowatt hour of power that's produced. So you don't want to add any expense. You'd like to put it into a panel very similar to what you currently stick on your roof, and you don't want to track it. So there's a rule in concentrators that if you have high concentration, it reduces your field of view. So you can't make this too highly concentrating, and actually this is only about a 2x concentration system here. But again, at the time, it worked pretty well. These are some actual systems that were made. These show the holographic elements here that were made on that roller system. And it was actually, there was a competitive advantage back when silicon cost about $200 per meter squared. However, currently the cost is is down to around $100 a meter squared. So with that, it kind of lost its competitive advantage. But it was a useful effort at the time and kind of opened the possibility for some other things. One of the best things about this was that they actually had this panel certified in their accelerated life testing, which allowed that panel to be certified for 30 years of operation without degradation. And anybody who knows dichromated gelatin, that material is extremely hygroscopic, and you get any water in there or anything, it will degrade. It will vanish. The hologram will disappear. So people ask me all the time, well, how well does it hold up? Well, here's an example of a panel that was certified, and it does show that you can use these kinds of elements in long-term systems. Okay. Another system that one of my former students, Deming Zhang, worked on was a what we call an ultralight trapping filter. So back in 1982 or so, Eli Jablonovich showed that if you put surface roughness on the top and bottom sides of the PV cell through a randomization process, you can increase the overall path length by a factor of 4n squared. Okay, so you say, okay, that's nice, but silicon has a refractive index of 3.5, so this is a factor of 50 improvement in the optical path length, which means that instead of making a concentrator, and reducing the amount of material aerially, now you can make the material really thin, right? If you can increase that optical path length and capture that light. Some other people over in Germany, Ulrich, and showed that there's another way to do this as well. You can actually enhance that further by restricting the escape cone for light coming out of that PV system, out of that PV cell. So you can do this, and they showed that you can do this with Rougate filters, which are very expensive and not really a scalable filter technology. So Deming and I looked at ways of using a holographic element. So for this, what we actually use is I think this other slide shows it a little better. So we use a reflection hologram here. The light at all wavelengths comes in and is scattered. Both the short and long wavelengths are scattered. But now when that light comes back to this holographic filter, it allows the shorter wavelengths to pass, but diffracts the longer wavelengths. All right. Now the advantage of that is that the highest conversion efficiency for a PV cell occurs right at the band edge or very close to the band edge. If you don't excite, you have a band gap, right? So if your photon doesn't have enough energy, you know that you can't do a transition. If it has too much energy, then for recombination and current generation, that excited electron has to decay down to the band edge and drop. So that decay is lost in heat. And Shockley Quasar showed the limits of that back in 1961. So what you really want to do is collect as much of the photons right near the band edge. So Deming made a prototype of this with a holographic filter and dichromated gelatin and was able to get a pretty significant, about a 15% enhancement in the absorption enhancement in that configuration over what you would get with just using Jablanovich's diffuser kind of scattering approach. Okay. All right. So now let's talk about another application here, and that is spectrum splitting. And this is one that we've been working on for a while because I think it has great potential. So what spectrum splitting is it's a multi-junction system. Most traditional multi-junction solar systems are concentrators where they have a very expensive 3-5 generally multi-junction cell, which is in tandem, and so you have one band gap growing on top of the next. You have a large 1000x collector. You focus that light down onto that cell. The reason for that multi-junction cell is like $50,000 a square meter. It's a very expensive PV material. So you want to make it a centimeter. And Roger Angel is very good about this. You know, in Renew, his company, they make these large collectors out of using their slump glass process and focusing it down. Disadvantage, and you know, the verdict's still not out yet on this, but the disadvantage is that in tandem, you have to be current matched. So the total amount of current that you're going to get out of that multi-band gap tandem system is limited by the weakest producing band gap. All right, just like your Christmas tree lights, if one goes out, the string goes out. The same would happen with that multi-junction cell. So here, we get rid of that by separating the cells. We can also use, we don't have to lattice match anymore. So this opens up, we could use silicon and gallium arsenide, gallium antimonide. We can use cadmium, whatever material we want. We can make a whole range of different systems. Now, so we shift the complexity from the cell to the optics. All right. So now the purpose of the optics is for this area that's over the blue cell. We have to redirect the red light over to the red cell, and the blue light that's over the red cell has to go over to the blue cell. Okay. And so these plots here show some different spectral response characteristics for different cells. Here's the gallium arsenide alpha cell, a silicon cell, and a gallium antimonide cell. So you see here that there are ranges that are obviously where you want to redirect the light, but all of these, an ideal cell would just be a triangle where this would come up here, and so there's an area of overlap here. So you have to have a way of evaluating the advantage of redirecting that light where there's overlap. So to do that, we've developed a set of metrics. The spectral conversion efficiency represents the spectral responsivity, which is akin to the absorption, the open circuit voltage, which is a reflection of the band gap, and the fill factor, which is a reflection of the circuit characteristics of the cell. And so we came up with a way of assessing this by looking at the efficiency of the spectrum splitting system, which is here, compared to the best cell efficiency in that group. All right? So we call this the improvement over best band gap, or IOBB. One of my former students, Juan Russo, wrote a nice paper on this in Optics Express. But this shows if we had ideal filters, represented by these rectangular plots here, we can get efficiencies on the order of 41.6 for this 4-cell system, and improvement over the best band gap, which would be this alta cell, of about 48%. Okay. These are some different configurations that you can have for the spectrum splitting. You can use reflection filters. There have been some systems that use dichroic filters, but again, dichroics are very expensive. But we can make reflection holograms that work pretty well. Another system, though, that we've been focusing on lately is this dispersive transmissive filter, and I'll explain that shortly. So one version of that, this is work by Juan Russo, where he just uses a simple holographic grating and a lens to form a dispersed power across the image plane here. You're shown this nice figure. And then in analysis here, using a 3-cell system here, was able to get, for an ideal case, about 39.3% efficiency with an improvement over the best band gap of 35.7. With an experimental hologram, the efficiency drops because, again, it's not an ideal square filter, 34.6, and an improvement over best band gap of 18.6. So, again, this wasn't an optimized hologram, but it does show that we are able to get significant improvements using this approach. So what this improvement over best band gap tells you, essentially, is how, what is the worth of this system? Okay. If I can make it with this more expensive cell, how does that compare in cost to adding this optical system, which is also going to add to the cost. So you can kind of weigh these two to figure out which one's going to pan out. Okay. One of my other students, Shelby Vordran, had another approach to this using a surface relief grating. She developed an algorithm to form what she called a broadband Gertzberg-Saxton algorithm to make a computer-generated hologram, basically, that would redistribute the spectrum correctly for a combination of different bandgap PV cells. In this process, it's basically an iterative process where you start out with what you want on the image plane here. You do a back Fourier transform to find what the phase relief pattern is here at the hologram plane. You then propagate and then see if it matches, and then you do kind of iterations here. You're able to get a pretty good filtering function. However, and she demonstrated this. She actually fabricated some through Tom Milster's fab process, maskless fab process that he has. But one of the issues with this approach was that the amount of, the light that came through had to be direct illumination. And even in Tucson, we have about 25% of the light, solar illumination that's available is diffuse over the course of the year. So that means that we're throwing away, and this is a problem with a lot of concentrating photovoltaic systems, is that they have a very narrow field of view, so they're not going to be collecting the diffuse illumination, which comes from a broad range of angles. All right. So we are looking at another approach, which actually Roger Angel came up with, and we've been working with him and implementing it, is to use a volume holographic lens approach, which focuses the light at a specific wavelength, what we call a transition wavelength, which falls somewhere in between what's needed for the wide bandgap PV cell and the narrow bandgap PV cell. The focus of this holographic element falls along an arc, and so light that is longer in wavelength will already come to focus and start spreading, so it illuminates the PV cell. And light at the shorter wavelength has not yet come to focus, and so it's also spread and illuminates the wide bandgap PV cell. The advantage of this approach, though, is that it can collect the diffuse illumination, because if it's designed for, this is also a tracking system, or needs a tracking system to some degree, although not as stringent as the very high concentration systems. So this is collecting direct light at normal incidence, but if light comes in at non-normal incidence, it's no longer satisfying that Bragg condition. There is some Bragg selectivity there, so that light will get through and illuminate the PV cells. So this just shows, again, we made some demonstration systems here, and this shows the sort of a plot of diffraction efficiency at different wavelengths across that volume holographic lens, which is actually pretty uniform for that element. And this shows the filter function. Again, not ideal, but again, it doesn't have to be ideal to show significant benefit in improvement over the best bandgap cell. So Ben Chrysler, who's here, has been working on a stitching process for stitching these cylindrical lenses together, and is putting together a kind of a demonstrator system using a Alta cell and a gallium arsenide Alta cell and a silicon cell fabricated by Zach Holman's group up at ASU. So this is part of a ERC engineering research center that we have with ASU, Caltech, Georgia Tech, MIT, a whole bunch of schools involved there. So let me conclude now with a few comments here. So again, the takeaway I want you to have from this talk is that holography is a very useful tool. It's been around a long time, but it's a very useful process that has a lot of applications because of the fact that, again, you can use these features, the selectivity, the multiplexing, the complex recording functionality, that like this Wavefront recording and so forth, all of these things are very useful in making kind of interesting optics. Now, another big thing about holography is the materials. So there has been quite a bit of improvement in polymer materials. One of the best commercial materials right now is produced by Bayer, or was developed by Bayer, now is distributed by Covestro. We use it in my class. If we have 5 experiments that we do in the class with that material, it's very nice. But it doesn't, for me, it's good, but it doesn't have the flexibility I need, because I need a lot of variation. So that's why we have our own material development work going on as well. Again, it could be used with this highly selective volume holographic imaging microscope. And on converse of that, it could be used in thin film, thin, very broadband kinds of collection applications. Now, my final point, I want to point out something else, and that is there's a real need for people with experience in holography for some of the new eyewear that's being developed, the augmented reality eyewear that, and it's a good job opportunity for a lot of people, because there aren't many schools that teach holography, but here again, you have eyewear. It's very thin. They're using, guided kinds of imaging systems. And there's a great competition for this, because this is a multi-billion-dollar industry that's going to form in augmented reality. And all the big players, Microsoft, Google, Apple, Oculus, and Magic Leap, they all have their feelers out. Because they need people in these areas. I went up to Microsoft this summer. I have a small project we're trying to kick off with them, and I've recognized a lot of students from my class were up there as interns for them. And Andrew Wall actually he thanked me for my class, because he said, and having experiments in that class, because when he went up for his interview, they asked him, how do I make a hologram? And so, here's how you make it, So anyway, I think holography, I know there's a lot more cutting edge areas in optics these days, but there's a lot you can do in holography if you're inventive and creative enough. There's a lot of really interesting things. It embodies a lot of optics. So, it's a good tool, if nothing else, to put in your bag of tricks as you move on in your careers. So thank you very much. Appreciate being able to talk to you.